Welcome back. We should continue with discussion of what is blood-brain barrier, and let's start with its classical definition, which is that blood-brain barrier is the continuous endothelial membrane of the brain vasculature which has sealed cell-to-cell contacts and is sheathed by vascular neural cells and perivascular astrocyte amphib. It functions to separate the circulating blood and brain compartments and strictly regulates blood to brain and brain to blood transport of solids. In order to show you more precisely, maybe in cartoon way and non-professional way, but what is it? Let's consider that some kind of bad virus is trying to enter our central nervous system. So it tries to get in, but endothelial cells have tight junctions in between of them so that this virus has no chances to pass. Let's consider another situation. It somehow is able to pass through, but here we have astrocytes, which actually are checking who is that one which is trying to get in, and if it says that it's someone else, so that go out of here is the response of astrocytes, so that these viral particles have no chance to pass. Unfortunately, that is not always the truth, and sometimes some infectious agents enter the brain through other route. They go through the periphery, they enter from nervous endings, get in through the retrograde transport, and eventually they reach the king, the neuron, so that we notice that the neuron disappeared here through some apoptotic mechanism. Another pathway is possible. If we have situations with increased permeability of blood-brain barrier, including stress situations, they get chance to come to our neural system and they destroy our nerves. Instead, they proliferate within our central nervous system. Much commonly, this is the case if we have immune-compromised patients, and like JC papillomavirus is an example of that. Whatever, we should really appreciate what is the blood-brain barrier, and that's why we have here a description of Goldman's experience, who injected tryptophan blue, which was a dye, into the blood, and the result was that both brain and corticospinal fluid were not getting this ink inside. In second experiment, the dye was injected into the corticospinal fluid, and the result was that brain also contained this dye, but not blood. So that the third C picture shows the conclusion that we have impermeable barriers between blood and brain and blood and corticospinal fluid, while we have a permeable barrier between brain and corticospinal fluid. It's actually important that these vessels that we see here as well as here, they all together result and they make up a huge distance of vessels. Imagine that it is 644 kilometers length vessels, and all of them are accompanied by this complex structure of the barrier. I want you to imagine how long is it. If you consider, and if you're familiar with Armenian events, our north-south road is 556 kilometers length, or distance from San Francisco to Los Angeles is 559 kilometers, while our total length of vessels in the brain is 644 kilometers. I agree, those crazy scientists that calculated this are real crazy, but nevertheless, we should appreciate how huge is contribution of astrocytes and other elements during whole this pathway to create enough impermeable barrier so that what is the role of this barrier? Actually, although representing only 2% of total body mass, our brain consumes 20% of our body's glucose and oxygen, and can rapidly increase blood flow and oxygen delivery to its activated regions. The process that we will call a bit later neurovascular coupling. And capillaries are the smallest cerebral blood vessels. They account for approximately 85% of cerebral vessel length and are a major site of the blood-brain barrier. In the human brain, capillaries provide approximately 12 square meters of endothelial cell surface area, which is available for transport of solids from the blood to the brain and vice versa. And the mean intercapillary distance in the human brain is as little as 40 micrometers. Therefore, solid equilibration is almost instantaneous throughout the brain interstitial space once molecules cross the blood-brain barrier. Actually, we should also appreciate the fact that except the blood-brain barrier itself, which we can see at the picture nearby, we have actually three barriers. At blood-brain barrier here we see endothelial cells with pericyte and astrocyte food processes. But also we have arachnoid epithelium, which is another separating barrier, as well as we have choroid plexus epithelium shown here, which also is producing cerebrovascular fluid. Currently, in order to understand the structure of the barrier, we identify different structures, participants of this barrier formation. It's only simplified way to consider that endothelial cells do that and astrocytes, but we should appreciate that local microglia as well as neurons have their contribution to regulation of local blood flow and permeability of the barrier, so that we can even say that endothelial cells and pericytes are separated by the basement membrane. Pericyte processes most of the outer side of the basement membrane. At points of contact, pericytes communicate directly with endothelial cells through the synapse-like PEC socket contacts. Astrocytic and food processes unsheath the microvessel wall, which is made of endothelial cells and pericytes. Resting microglia have a ramified shape. Here are the rami. In case of neuronal disorders that have a primary vascular origin, circulating neurotoxins may cross the blood-brain barrier to reach their neuronal targets, or pro-inflammatory signals from the vascular cells or reduced capillary blood flow may disrupt normal synaptic transmission and trigger actually neuronal injury. This arrow number 1 shows this ratio, this relationship from vascular damage tissues to the neuronal injury. Microglia recruited from the blood or within the brain and the basal wall can sense signals from neurons, so that if neurons signal that something is wrong with them, the signals come here to the pericytes, to the barrier structures, as well as to the microglia. Activated endothelium, microglia, and astrocytes signal back to neurons. And the result is that usually it could aggravate neuronal injury. In the case of primary neuronal disorder, signals from neurons are sent to the vascular cells and microglia. Here is 2 numbering codes from neuron to astrocytes and pericytes, etc. And then this activates vascular glial unit and then contributes to progression of disease. So signaling back is shown by this 3 arrow. To the right, we see another schematic representation of the fact that coordinated regulation of normal neurovascular function depends on the vascular cells, we mean endothelial cells and pericytes, as well as neurons and astroglia. So facts like blood flow regulation, microvascular permeability, cell matrix interactions, neurotransmitter turnover, angiogenesis, neurogenesis, they work coordinated and together. That's the idea of providing such an idea as neurovascular unit. Moreover, the more we understand the function of this barrier, the more we appreciate its role in neurodegenerative disorders. An example is shown here, role of the blood-brain barrier in Alzheimer's disease. And we can follow the events and in early phase, brain hypoperfusion and impaired blood-brain barrier clearance of amyloid beta lead to accumulation of neurotoxic A-beta oligomers we mean, in the brain. Reduced blood flow and accumulated A-beta oligomers can both initiate the neuronal injury. In the early symptomatic stage, we have much more pronounced reductions in the blood flow, activation of endothelial and pericytic cells. We have loss of clearance of A-beta proteins, and we have increasing accumulation of A-beta in the brain, so that microglia and astrocytes should consider this as a huge problem, so that neurofibrillary tangles also accumulate in neurons in response to this ischemic injury due to decreased blood supply and A-beta accumulation. Then in the late symptomatic stage, we see much more A-beta oligos here, and degeneration of endothelial cell wall and pericytes precludes clearance of A-beta and eliminates the blood flow from the capillary unit. This results in accumulation of metabolic waste products, changes in the pH, electrolyte imbalances, etc. These chemical changes in brain microenvironment present an important challenge for synaptic and neuronal function. Amyloid actually accumulates on the basal wall. There is also a prominent accumulation of intraneuronal tangles. At the end stage, we have profound neuroinflammatory response and the collapse of capillary unit. Actually, you can see that there is no unit here. It has collapsed. It's not functioning. It's not actually there. And this is accompanied with the loss of axonal dendritic synapses and neurons, which disappear under amyloid deposits. The main idea here that we showed was developed by Zlokovich 10 years ago, but some updates to this are found here, so I provide you a link if you are interested to go ahead and check out what are other updates to this process today. So to conclude, normally blood-brain barrier is quite impermeable for lots of substances, but at the same time lots of substances just have no chance to pass blood-brain barrier. Yeah, water-soluble agents may pass through the tight junctions, but many other water-soluble ones have no chance. Alcohol, water, okay, others, no. Well, lipid-soluble agents, including drugs, may pass through, and that is an important point in neuropharmacology to decide whether a given drug or substance is able to pass or not. Some other substances like glucose, amino acids, they pass through by help of special transport proteins. Additionally, we have receptor-mediated transcytosis mechanisms. Large proteins such as transferrin, insulin, low-density lipoproteins, leptin, immunoglobulin G, IGF, they all pass using receptor-mediated transport systems. There is another one for molecules like albumins, which require so-called adsorptive transcytosis, so that those who really are required for the brain, they will find their way to brain. Those which are not needed, they wouldn't pass. But pathogenic factors may hijack either of these mechanisms to pass to the brain, which is not expecting them at all. So we conclude this short introduction, and let's move to pathogenesis of nervous system disorders. And some of mechanisms of neuronal injury are really non-specific, means every cell has the same mechanisms of damage, which we mentioned in early September, so that mechanisms like ATP deficiency, membrane damage, impaired protein synthesis, impaired signaling, electrolyte imbalance, apoptosis dysregulation, all these are mechanisms for any cell disorder, and this happens in the brain and neuron as well. But in neurons we have some specialized structures which create additional mechanisms of specific damage. It includes problems with neurotransmitters, decreased or increased their production and their outcomes, impaired axonal transport, impaired functioning of synapse. Very interesting mechanisms which we will touch together, demyelination, and excitotoxicity, which, as we mentioned, is an important mechanism in such disorders as ischemic brain disease with stroke, traumatic damage to the brain, epilepsy, Alzheimer's disease, and even demyelinating disorders as multiple sclerosis. So that let's, keeping in mind these specific and non-specific mechanisms, briefly go through the main groups of nervous system disorders. So some of them develop as developmental abnormalities like ageria. We see that there is no gyrus here at all. It is flat. Some our colleagues argue that such kind of brain structure have very intelligent police workers, and that they only one goes here and another here, which is the site where the head of this worker is localized on his skull and therefore has imprinting in the brain. But of course this is just a joke, but let's really consider the fact that whole brain is white. Very few gray matter and there are no gyri, showing that it's underdeveloped brain tissue. Another developmental problem could be hydrocephalus. Let's be cautious. When we say hydrocephalus, it means that we have increased volume of ventricles, at least in this case, or in general of cerebrospinal fluid, which might cause compression from inside towards the nervous tissue, and we have a huge volume of ventricles here. It could be a problem of impaired resorption of the cerebrospinal fluid. Some obstruction could cause this, as well as developmental problems. But this specific picture is shown as a problem in development. Next group would be infection, and of course different infectious agents might cause damage to the brain. Despite the fact that we have a blood-brain barrier, some of them pass, as we discussed, using their own ways. And even our protective mechanisms like meninges are sometimes becoming the site of a problem. So here we see a picture of meningitis, which is the reason why neurons themselves even may not be affected yet, but it's enough to have severe meningitis to have even lethal effects. Prion diseases are separated from infections because of their unique biological nature. As you know, about 20 years ago or so, Stanley Prusiner got Nobel Prize for showing that there is a separate group of diseases which is actually infectious, but those causes are not related with any microbe because there is no altered foreign DNA to cause it. The problem is in specific proteins which are called prion proteins. In prion diseases we have quite another mechanism of action. To exemplify its mechanism, let's consider that PRPC is the normal protein, while PRPSC, SC shows scrapie protein, is the abnormal beta sheet containing protein. And actually the problem is that the scrapie proteins have ability to come in contact with the normal cellular proteins and turns them into scrapie forms, so that after some time and after some interaction, all of normal proteins become scrapie ones. And these abnormal folded proteins cause damage to the cells, and so they actually degenerate. An important feature of this disorder is that it happens quite fast and it is able to pass from a cell to another one, so that the initial start is slow, but then it is developing in accelerating mode, and more regions and more cells get affected. Pathologically, the picture resembles that of a sponge, so that this encephalopathy was called spongiform encephalopathy, and it is a common problem in animals and sheep and cows, so that scrapie, the word comes from similar problem in sheep. We have had a story of mad cow disease, which was very important and considered as a dangerous situation about 15 or 20 years ago. And we have disease in human as well, known as Creutzfeldt-Jakob disease, in which within weeks to months, very fast degeneration results in memory loss, loss of ambulation, and eventually death of a person. It's incurable disease today, but the the idea of understanding the tiny mechanism of disorder is still promising. Maybe one day we will be able to understand how to cure it. In some populations, like in Papua New Guinea, there was a tradition that when one of the famous representatives of a family died, the relatives were eating his brain, and they were hoping that his strength and power would come into them. But actually they were engulfing scrapy proteins, which, another mystery, were passing through the gastrointestinal system to the blood, and then they were entering brain, and they were altering whole brain structure. So this group of disorders are known as prion diseases. Let's continue with cerebrovascular disease, and frankly speaking, we have one of the most important killers in the world being stroke. And here you see the major two forms of stroke, ischemic and hemorrhagic. In hemorrhagic, we have damage to the vessel, so rupture of vessel results in a local damage here, and it accounts for about 15% of all strokes. But much common stroke is ischemic one, where usually a thrombus is occluding an artery, resulting in ischemia in corresponding region, and the neurons that die, they result in corresponding change in clinics. Means we may have numbness, we may have paralysis, we may have sensory or motor deficit, so that ischemic stroke is one of the leading problems in the world now. And of special importance is that here we have a very huge chance of prevention if timely and accurately diagnosed, and if we are able to remove this blood clot in time, we might have survival of neurons. About this we'll talk at the end of our today's lecture. So that separate problem that we're going to discuss today, we protect our brain with perfect skull, but nevertheless traumatic brain damage remains an important problem. And if the mechanical factor is severe enough, we may have different types of damages in the brain. Sometimes it's really important to differentiate whether this hemorrhage happened above or below the dura mount, so that if it is above it, usually we have accumulation here of an arterial blood, and due to, let's say, skull fracture, this bleeding results in so-called epidural hematoma. And this is opposed to subdural hematoma, where we see the skull itself is not damaged, but actually bleeding happened. Means we have subdural hematoma, and here venous blood usually accumulates and it directly compresses the brain tissue. There are important differences in these two, and the management also differs. I just want you to have general understanding of what kind of hematomas we should have here. Next problem is demyelinating disorder, which will be a topic of special discussion, so I wouldn't click here on anything. Then we pass through neurodegenerative disorders, examples of which include Alzheimer's disease, Parkinson's disease, Huntington's disease, system atrophy, lateral amyotrophic sclerosis, etc. What is common for all of them is that they are chronic, they are progressive, the mechanism of selective neuronal loss are not fully understood, but usually when we have neurodegeneration, it means that most prevailing mechanism is apoptotic cell death, and variety of neuronal cell injuries contribute to more or less selective death of neurons. In case of Alzheimer's disease, which is within the 10, it is usually the 6th or so in the developed countries cause of death. And unfortunately, it is also tragic by its natural progression, because being a disease commonly affecting elderly people, they lose their memory. And if we think deeper, what we are as a person is our memory. Who are we without our memory? We are just organisms. That's why sometimes, maybe not a good term, but some people call it a vegetable. It's not actually the same term here as applied to those with Alzheimer's disease. But if someone is losing his personality, it's a huge question, who is he then? On this picture, a patient died from Alzheimer's. We see that there are very deep spaces in between of these strata. It shows that the gray matter, the neurons, are affected. Most affected ones are also those of hippocampal structure, which is responsible for most of our memory. Next group of disorders includes different hereditary metabolic issues, as well as acquired metabolic issues. So this point 8 is different metabolic issues. It includes hypoglycemic problems, because we know that neurons are selectively dependent on glucose blood supply. It involves problems with hepatic encephalopathy, because we know that ammonia preferably affects brain, causing hepatic encephalopathy, different electrolyte disorders, etc. So this is group of metabolic issues. Separate group involves different vitamin disorders, like B1 being important in causation of Wernicke-Korsakoff syndrome in alcoholic patients, or B12 known as subacute combined degeneration of spinal cord. And we remember that especially lateral and dorsal tracts are being affected selectively with sparring motor pathways. Separate problem are tumors of central nervous system, and here we see the relative frequency of them. The intriguing fact is here the fact that neurons usually are not proliferating at all, so one could not expect any tumor within the brain. But actually we should not underestimate that we do not have only neurons in the CNS, but we have different other cell types, including glia, including vessel cells. That's why the most common ones are meningiomas derived from meninges. We have different tumors derived from glial cells, and that's why glioblastoma is the next most common. We have an endocrine organ here in the brain, which we discussed in more details in endocrine system disorders, pituitary tumors, and as well as many others. So we here would conclude the main groups of nervous system disorders, and we'll talk in a bit more details about some specific forms of nervous system disorders.